Yeah, well, that's Reinhardt's little joke. But actually, I don't mind it so much, to tell you the truth. So, look, I want to thank SPS for this. I'm really honored by it. I've had a lot of, you know, good things happen, and this is certainly one of them. And one of the things I really actually owe more to the SPS than the SPS owes to me, because life has been really pretty darned interesting as a result of safety pharmacology, for me at least, and probably it sounds like for a lot of the people in this room. And the other thing that I'd say is that, you know, what I'm going to talk about is really pretty much derivative from the electrocardiogram. And I'm really like the appropriateness of being able to talk about it in a country where Eindhoven 110 years ago invented the first practical ECG. So, you know, what's really amazing to me is that after all these years, this cardiac safety stuff is still front and center. Amazing. Either it's really complicated, or either we're really stupid. One of the two. Probably a little bit of both. So S7B, as you know, started in '98. The final concept paper was in 2000, was adopted in 2005. Long process. E14 wasn't nearly as long, which tells you about the relative impact of clinicians versus non-clinicians. It took them really about a year and a half to get E14 adopted. So the results have been, and you've heard all of this, and a lot of it, I think I can fly through some of these slides because Gary and others have really talked about this already. But you know, what's happened is there's very clearly been a reduction in post-marketing phase 4 reports of Torsades de Pointe from non-antiarrhythmic drugs, which I call NARDs. And there has been a continued approval of drugs with QT risk where the benefits are greater than the risk, and I speak particularly of oncology, but other areas along those lines. It was very important to understand that I certainly believe, and I'm going to show you examples of this, where candidate selection has really been compromised by false negatives and false positives. We've heard a little bit about this. A cottage industry sprung up as a result of E14. I mean, you know, if you got 10 cardiologists together who could read an electrocardiogram, they formed a company. And you wound up with what turns out to be $1 billion business, measuring TQT at an incredible level of sensitivity. There's been a lot, I think, of drug mislabeling. We've heard about that, and I think that can be attended to. So anyway, the upshot of all of this is that cardiac safety is still a big item. And when I counted it up, it looked like there was more than 50% of the platform presentations, which I had access to, concerned cardiac risk assessment. So look, what I'd like to do is give you an idea of my own trajectory in this field, because I think it also makes a few points that maybe you can get. When I started, it was in 1990, and there was a report by Brian Monahan and Lou Cantilena and others in that group, who showed that terfenadine, which was the first of the second generation antihistamines, prior to that, the first generation, people were dying from car accidents because they fall asleep. The thing about this one was that these guys didn't cross the blood-brain barrier, so you got rid of that problem. And it made Seldane, or terfenadine as it was known, a very popular drug, and it was selling like crazy. It was a blockbuster drug. When I started in it, one of my postdocs, Dave Rompey, when I was at Baylor and Texas Medical Center, was working for Mary and Merrill Dow, which survives to this day as part of Sanofi Aventis. Now, we were looking in those days at the outward potassium current in guinea pig cardiomyocytes, which was responsible for repolarizing the action potential. But at just exactly that time, Mike Sanguinetti and Nancy Jerkowitz, who were at Merck, did really a beautiful paper in which they were able to sort out biophysically and pharmacologically the rapidly repolarizing potassium current called IKR and the slowly repolarizing current called IKS. So now we had gone from knowing this overall current to specifying these currents. IKR, I still regard that as one of the really, really neat papers, because you could see with IKR where sanguinity was going with this. It was a really unique channel, because what happened with that channel is that it had inactivating, rectifying properties that were faster than the activating properties, which makes it very, very important for being sensitive to changes in voltage. I don't have time to go into all of the details, but it's really a beautiful channel for regulating, being important in regulating the duration. Anyway, let me go back. So then, let me just move along here. Ray Woosley and Rich Gillis looked in cat, because it turned out that cat they were lucky, that cat had a lot of IKR, and they popped terfenadine on there, and sure enough, this IKR, which had characteristic properties, I'll show you more later, that was it. It showed up, and it showed up at the exposure level you might expect. So that was really a very good experiment. Joel Morgenroth and I did some work trying to look at comorbidities in the clinic. And then Dave and I took a look at terfenadine and metabolites. It was a Merrill Dow drug, and we knew a lot about the metabolites. And since we had IKR and we could work with it very well, we ran all the different metabolites through it. It turns out actually there's really a key metabolite which shows up because terfenadine is essentially a prodrug, and in one pass through the liver, it gets carboxylated to terfenadine carboxylate. It's just an important part of the story. Now, I was very interested early on in the notion that you could have polypharmacy effects. So one of the things that we were twigged to very early, Dave and me, was the role of verapamil. So verapamil, it was in the clinic. It was a calcium channel blocker, but there were reports out there that suggested that it could actually change, increase the duration of certain aspects of the action potential. And Dave and I went in there and looked at it on IKR, and sure enough, it was a very potent blocker of IKR. Okay, so then things got really much clearer in 1995 when genome association studies for long QT syndrome done by Mike Curran and Mark Keating and others in that group showed that there was a link between what was called LQT2 and hERG. It was actually, there were mutations in hERG that were responsible for LQT2. And then Mike teamed up with Mark Keating's group, and what they showed was that, because he'd known all of this stuff, that the hERG channel produced the IKR current as he had characterized it both biophysically and pharmacologically. So that was really a terrific move that now let us work with molecular genetics and moved us away from, and gave us a lot more specificity in what we were looking at. I think the other thing, probably, I don't know how many of you know how this name came about, but there was a Drosophila mutation in something called the ether agogo gene, which when the Drosophila was exposed to ether, its legs shook, okay? And so then they found in humans, Warmke and Ganetsky, that there was in fact a human homologue of this, and so that's how you get the name, human ether agogo related gene. And I'm talking really about the gene product, which is IKR. So then it didn't take, it didn't take much for Mary Louise Roy, Robert Dumaine, and me to express now the messenger RNA. You can express it as cDNA if you like, and test whether or not the target really was hERG. So you inject the stuff in a cell, and you get tons of the channel, and you do dose response curves. You look at exposure and all that stuff, and sure enough, it all came out right. This was published in Circulation. And there's a really very interesting commentary by Mike Rosen, who was in pharmacology at PNS in those days. And Mike pointed out that, this was really the tip of the iceberg, that there were antibiotics and other kinds of drugs that had been associated with torsade de Pointe, and that we ought to be keeping that in mind. And sure enough, the story evolved exactly that way. So then, we did some stuff on dofetilide. Dave and I did cisapride, which was very popular prokinetic drug for heartburn. We looked at ketoconazole in '98. Then we did I told you it was a prodrug. Fexofenadine is terfenadine carboxylate. Craig Pratt and I did some stuff on clinical safety. And then the sort of thing that sprung out of the work that Derek alluded to before, which was these ondansetron, granisetron, these 5-HT3, which is a ligand-gated channel, which were used as antiemetics. We did some stuff on that. And the interesting thing about those guys was that they prolonged QT, but they did it by changing QRS. It wasn't a big deal. It was interesting. And one of them actually popped up as being a hERG blocker. Now, at this time, vanoxarine showed up in my life. So we tested vanoxarine in the lab. This is a very interesting drug. You're going to hear more about this as we go through this process here. So vanoxarine had been in the clinic in man. It's a dopamine transporter, highly specific blocker, very, very potent. And it had been used for treating depression and Parkinsonism, but it had no efficacy, but it had been in man. And so we tested this drug, and it was a potent hERG blocker. So how in the heck could we explain the fact that here's a potent hERG blocker, gave no signals in the clinic? And so right away, the old verapamil story comes back, and so we started to look at other channels like calcium and sodium. That's the theme I'm going to be talking about throughout this talk. We called them multiple, actually it was Laurie Hansen who gave them this name out of one of these Ilse Hesse papers. So multiple ion channel effects, we call them mice. And then mice crops up all the way through here. Then Ficker and me and others in the lab, we got interested in hERG maturation, showed that there were chaperones that did it. Then it winds up, the delayed effect of arsenic. Here Agatha Christie has them on the Orient Express, and they all drink tea that night, and the next morning one of them's dead, So you have a delayed effect, and this delayed effect is due to block trafficking of hERG. The lifetime of the hERG channel is roughly about 16 hours. So if you can prevent trafficking, then you've got this nice little time sequence that you can escape. You can you can get away with, but don't try it on your. The cops are wise to that. Then we did pentamidine. That was an interesting one. And so we got very interested in trafficking, which are these delayed effects, which I talked about. Then we did a little stuff on alfuzacin, which is an antiadrenergic blocker. It's used for urinary retention. And that one got us interested actually in the late sodium current and its effect on QT, and I'll talk more about that later, some stability stuff. Then, vanoxarine really began to come front and center in our lab. We worked out the mechanism. We took it into, and I'll tell you more about this later. We took it into a very good animal model. We got involved with stem cells. We'll talk about that. This methadone diazepam interaction stuff was, again, multiple ion channel effects that could explain that stuff. Vanoxarine, AF reinduction. Then Dave and I just published in JPTM this historical paper, which I think is, it captures much of the flavor here. And then we had a paper in Nature Scientific Reports very recently on multiple ion channel effects. I'm going to spend a lot of time talking about that. I should tell you that vanoxarine is looking awfully good, and I will say a word more. I will say a word or two more about that too as we go along here. Okay, so I can sort of I think I can slide through these slides pretty quickly. This is the culprit, twisting of the points, and there's no effective heartbeat, so you faint, and 1 out of 5 times you die. This is the standard hERG current. These are these incredible properties. So when you activate the thing with voltage, you get these increased currents, and then all of a sudden at higher voltages they actually get small. So at high potential, the thing actually rectifies and shuts down. And if you expose it to Seldane, you can see that you reduce these currents. Now, what's very interesting is this thing called the tail current. So when you repolarize, because the inactivation is very fast, it gets removed very quickly, and activation is relatively slow. So you get this really terrific waveform, and it's very sensitive. So you jump up here because you've removed the inactivation that was here, and then because activation itself is slow and deactivation is slow, you get to follow this thing. So it's really a slick thing. And so then, this is the way you really want to do it now. I think we've learned to do it. There are other protocols. The only thing that I think is very, very, so this is just the old story, that. What's really important is that since you're going to be constructing a concentration response curve, those curves have to have the K on and the K off for your drug, and it's almost all K off because almost all of these cases are diffusion limited. K off is important. If you don't get into a steady state, your concentration response curves don't mean a damn thing. And if they don't mean a damn thing, your IC50, IC10, IC20 doesn't mean anything. So this is the way we do it. And we say you're in a steady state when you've got a less than 2% change. I mean, you guys can decide whether you want it to be less than 1%, whether you want it to be less than 5%. It doesn't really matter in terms of trying to collect lots of data and having a good success rate. So that's the way you do it with manual patch clamp, which is the gold standard. So we published a paper in JPT, I'm Glenn Kirsch, that is, I don't know, seems like a long time ago. And here's some culprits down here, good guys and bad guys. And our data is shown here with the circles and the error bars. And so within the circles include the mean and the 95% confidence limits. And you can see that the data is really very tight. If you look in the literature, however, you can see that there's data that differs from the data that we had with manual patch clamp by a significant amount. And I'm not sure what the reasons for this might be, but I do think one of the important things is being in a steady state when you're making this measurement. And then over here, I want to show you, Gary alluded to the fact that if we're going to do this, I'm committed to it. I don't know whether SEPA is or not, but I can tell you I am. So then you have to know how good is your instrument. So we chose to work with an instrument that made gigaseals, just like you make with manual patch clamp. But these automated instruments, you have holders that carry the business end, which is glass, and these holders have plastic in them. And so hydrophobicity becomes a really important consideration. So for example, we used a particular instrument, the Q-Patch, high throughput instrument, and these are the values we got for these drugs. And so you can see that there's pretty good correlation for some of them, but some of them like pimozide and terfenadine, which are really quite hydrophobic, there's loss. So you're going to have some offset for those, but you can live with that offset if you have some idea as to what log P might be, if you have some idea of what an appropriate control to put in your automated plate to do this kind of thing. So anyway, so one of the neat things was that even at Marion Merrill Dow, I mean, we did, they got hammered because of terfenadine. No preclinical signal to speak of in those days, no effects on APD or in vivo. There were 5 cases per million patient months. The frequency is about 1 in a million, undetectable in clinical trials, human QTC surrogates. That's where it came from. And so the dosage was 60 mg BID, and you had a 2% change. And that's, by the way, I think one of the reasons why the TQT is has that upper 95% confidence number for the delta of around 8 milliseconds, which is a hell of a number. Then what we did is we looked at FEXO, and if you look at the normalized herd current as a function of concentration, you can see here's the story for terfenadine, the parent, and here's the real drug, which is terfenadine carboxylate. It's been carboxylated down in these positions, and it doesn't do anything. And so, Seldane went south, and fexofenadine or Allegra came on the scene. Okay, so then it was pretty easy to say it's all about HERG, even though we knew it wasn't. But a lot of drugs, and these drugs, by the way, they have no mitigating factors in terms of their effects on other channels. They just blast HERG. And so these were all, at this time, it seemed like there was even an epidemic of this stuff. And these drugs were pulled for these reasons, and then you wound up with the S7B, integrated risk assessment. The big boy in here was the HERG assay. But in our own data, and I think other people have seen this, if you do a 2x2 contingency and you look at you know, a hERG signal and whether or not you see an impaired repolarization, in this case with APD or QT, and a minus hERG signal. So you can see that there's, you know, for the data set that we used of 118 drugs, there was concordance for roughly 75% of them. hERG signal, which was present in a small number of them in this particular data set. You did get an APD QT signal, no hERG signal, no APD QT signal, but you had no hERG signal, and you would have impaired repolarization, or you would have the false positive. That is to say, you'd have a hERG signal but no impaired repolarization. So it's very clear it's not just about hERG. And we knew that. And you've seen this slide that Gary went through. Let's see. I just want to spend a minute or two going over this. This is a really critical current. IK1 is on at rest, and when you depolarize the heart, that current, and it regulates resting potential. When you depolarize the heart, that current shuts down, tight as a drum. And between that guy shutting down and IKR shutting down, you wind up having a membrane resistance during the plateau that is greater than it was at rest. And what this means is that tiny little currents, like the late sodium current or like the delayed calcium current, have outrageous effects on duration. That's something very important to keep in mind. So then here comes INA, and it doesn't show up well at all here, but there's a little bit of sodium current flowing out here that's having a hell of a big effect before the IK1 starts to come in. Here's the calcium. You saw that with Gary's. IKR, which, as I indicated to you, it has this feedback thing, okay? So it's shut down. Now you start repolarizing the membrane. That baby opens up because the inactivation is removed and current starts to flow. The more current that flows, the faster it repolarizes. So, and then let's see, there's a whole bunch of other guys I'm not going to go through. So we're working along here thinking that, you know, multiple ion channel effects are important. And, you know, we had the verapamil story, and we had this venoxarine story that was brewing in my own lab. And so we decided, and this is work that Jim Kramer, Carlos O'Bahara-Paz, Glenn Mite, Yuri Khrushchev, Glenn Kirsch, and other people at our company did. And I just talk about it. So what we decided is, you know, we're going to look at the three front runners. And what we did was we went and scoured the literature. And we were able to find, we've actually done 100 drugs, but we wanted to find drugs. We think the problem is not QT, the problem is arrhythmia. Nobody dies from QT. You die from arrhythmias. And so we looked at the outcome variable as TDF, TDP, Torsades de Pointes, and we used 3 criteria to figure out whether we annotated each drug in this 55 data set, 33 of which were torsadogenic and 22, or it might be 32 and 23, I can't remember, were pro and were plus, and I think 22 or 23 were not. So we used the Redfern paper to come up with TDP annotation. We used the Arizona CERT to help us with that, and we used the package inserts. And so we had to have a consensus. So even though we've done 100 drugs, we only wound up with 55 drugs that we were confident about what the outcome was, or at least reasonably confident. We have done a sensitivity analysis, and that we're still poring over that data. So then we said, okay, well, here's the 3 players, and what we're going to do, we're going to use automated patch clamp to get the throughput, because after all, we're doing 55 drugs, 4x3 concentration response curves. That's hundreds and hundreds of data points, and so there's no other way to do it. Here's what the data looked like against the drug number for IC50s for hERG, and again, the circles represent the 95% confidence limits and contain the mean values in there. And we did the same thing for CAV. Data gets a little bit out of hand up here, and also for NAV out here, and that is because for some of these drugs, you really can't the solubility limit is such you can't push enough in there at these high IC50s. And so there's some variability, a little more variability than we would like, but that's life. So then those guys had the smart idea that let's look at what these things look like. So if you look at the ion channel IC50, if you compare terfenadine and verapamil, and you're looking at the effective therapeutic plasma concentration and the IC50s for HERG, NAV, and CAV, you can see that for terfenadine, the block is quite potent, and it's certainly very near the exposure level, but there's no benefit. These guys are from CAV and NAV. So, the idea is that, if you got something that's prolonging repolarization, if you drop down the depolarization contribution to the repolarization, you can get these offsetting effects. And so there wasn't going to be any benefit from them here. But with verapamil, it's quite the different opposite story, as we knew. So here is the HERG IC50. Behind it is the CAV 1.2 IC50. They're right on top of each other. And here's the exposure. And so you can see that, if you do what we call V-plots, these are safety V-plots. The ETPC index is really the safety margin. So if you do these V-plots, you can see here's terfenadine, the HERG's down here, and the limbs are up there. And here's verapamil, so you get this V thing. And here's like sort of an inverted V. The verapamil is actually a more potent calcium channel blocker than it is a HERG blocker. So now if you look at the 32 torsatogenic drugs, and you look at the V factors, and then I don't think you can see it, but oh yeah, it doesn't show up at all here. But there's the Redfern cutoff line is around here. It's at 30 is this safety margin. So if the IC50 over the effective therapeutic plasma concentration has a ratio of 30 or less, that drug is dangerous. And so you can see that there are a lot of drugs down here that, that's right, they are dangerous because the level is up around here. But you can see there are some drugs up here that you wouldn't have thought were dangerous because they fall well above that 30 number, but they got long limbs on it. And it turns out that these drugs can be torsatogenic, probably because they don't, but we don't really know this. They don't get the benefit of this multi-channel block. On the other hand, you can take drugs that are clearly below the 30 line, okay? Like verapamil is this one here. And you find a bunch of these drugs with inverted Vs. That is, the calcium channel block is just greater than the HERG block. And so it offsets it, it mitigates it. I don't want to get into the details of why calcium may do it, because frankly, I don't understand. I have to have Carlos explain it to me every time. So, and then you get all the way out here, and there's no worry at all anyway, because the HERG block is not very much, and the CAV block is greater. Okay, so then we decided, okay, let's just look at the fraction of torsad per decade of drugs. So we looked the drugs in the list, the 55 drugs, 10 at a time, and we looked at the fraction of torsad as a function of the HERG safety margin alone. And the data are shown here in these circles. And another way, we wound up using a bunch of different models, and I'll get to that in just a minute. But another way we could plot the data, which came out of actually the models, but this is just plotting the data non-parametrically. And now we're plotting, instead of against the HERG IC50, we're actually plotting the ratio of the HERG IC50 and the CAV IC50. And this is the raw data. And you can see that here there's a sort of a gradual merging, but here there turns out to be, that is to say, as the safety margin goes down and down, the danger goes up and up. And here's ones where there's not much contribution, or any, if any, from CAV block, so the danger is there, but then all of a sudden when CAV starts to kick in, which is around this 0 point, which means that the ratios are equal, 10 to the 0 is 1, so you get this, you see this breakout like this for the fraction of TDP. So when you have this kind of thing, I mean, this is suitable for logistic regression analysis, and we wound up doing logistic regression analysis where we parameterize for the beta sub-zero and the beta hERG, using the safety margin as the argument. And then we had a bunch of models, and I'm going to really talk about model 1, which is hERG alone, and model 5, which looks at, and is the easiest way to look at, interactions of CAV. And it was the one that actually gave the least, the fewest misidentifications. And you can read all about this online at this Nature paper. So then if we just looked at it again, now what we did, so what we did was we had this training set of 55 drugs. Now how the hell could we get a test set out of that? So we did this leave one out business where you leave a drug out and then you run through the logistic remodel probabilities for the remaining drugs, and from that you then calculate the probability of the drug you left out, and you start, you have a lookup memory where you do this one at a time and you put the new, now cross-validated probabilities in another column. And so now we're looking at cross probabilities, and we're looking at torsadogenic risk from 0 to 1, and we're looking at model 1 and model 5. And you can see that for model 1, which is hERG alone, and you're looking at the non-TDPs and the plus TDPs, and there, there's not really a very clear separation. If you look at this model 5, which is looking at the CAV interaction with the hERG, you can see that there's a very, very clear separation. Here are the means, here are the confidence limits on either end, and here are the means here, and the usual quartiles, et cetera. So then we compared them with ROC curves with the sensitivity, which is the true positives over here, and the 1 minus spec, which is the false positives over here. Here's the line of identity. So if you scrambled our drugs, you couldn't segregate them at all. So if you look at model 1, this is what the ROC curve looks like here. But if you look at model 5, you can see that you get a much better ROC curve. So it's victory by ROC curve. And what this tells you, these arrows point to that position in the readout table where you have the optimum distinction between false and true positives. And you can see that the arrow here is displaced far to the left of the arrow here, which means that this does a better job of segregating true and false positives. The other thing that's really important, and I think this does reflect, it's very clear in this, if you want to get up to a sensitivity of around 90% or thereabouts, if you're doing hERG alone on this curve here, to get to this point here, a sensitivity that's approaching 1, you have to get something like 60% false positives. Now that's a big number for this particular data set, but there's no question about the fact that the overemphasis on hERG has resulted in a large number of false positives. And as a matter of fact, you cut that down by bringing in, for example, model 5 with this calcium offset, so you get to the same point here, with 1/3 the number of false positives. So if you do a contingency table and you look at the classification between model 1, hERG alone, and model 5, which was the best one, you can see that here you had roughly a 41 out of 55, which was 75% correct. But when we did it, we lost. You can see here that the false positives, or the false, which one is which here? Anyway, you can see that one went from 6 to 1 and the other went from 8 to 4. So there was a terrific reduction in both of these. And so now we've got, we're getting 50 out of 55 right. So we're doing it right 90% of the time versus 75% of the time, which is what we, was not unanticipated. So just summarizing here, the in vitro has about 75% accuracy in this data set that we used, the training set and the test set. Type 1 errors are most frequent, but type 2 errors occur. We propose the mice assay as a better predictor. We measure the concentration responses for 32 and 23. We used automated GigaSeal because we needed it for throughput, accuracy, robustness, and cost, a very important consideration. And we used the binary outcomes. The predictor variables were these logistic regression models, and the outcome variables were TDP. And so model 5 was significantly increased this predictivity. And the mice turnaround times are 10 to 20x faster than manual patch clamp. And the cost is really de minimis compared to, and I'll talk a little more about that as we, how am I doing here? Anyway, so the idea is, if you look at more than one channel, you can avoid unexpected outcomes. I want to talk a little bit about the drug that we would have tossed in the wastebasket based on HERG alone. And here's our clinical development program. So the non-clinical one, I've talked about, but we did things like the wedge deal. There was no transmural dispersion of repolarization. There was no reverse use dependence, all bad things to have. The mice hypothesis, one of the things that really came out was that, if you just looked at the IC50s at the usual pulsing rate, there was some separation between HERG and CAV. But once you started pulsing it and you started looking at use and frequency dependence, that CAV thing was just tremendously frequency dependent. The drug really was hard, does not come off a rapidly beating calcium channel. And that was very important because that moved it towards HERG. And that gave us the clue that, this thing could be very useful in circumstances where the heart rate is very, very high, and that's AF, atrial fibrillation, atrial flutter. So we went into an animal model. Sure enough, it worked. We've done phase 2A, phase 2B, dose ranging. We just finished it now, 200 to 400. Not a single serious adverse event for this potent HERG blocker. Primary endpoint was 1 minute of normal sinus rhythm. These were, by the way, the clinical trials. These are oral, they're double-blind, and placebo-controlled. So, the dose-dependent frequency of termination at 8 hours was 50%, 54%, and 76%. So that's a really, really good response. And like I said, this is a drug that would have been in the dumper. It wouldn't have even been considered. Now, this is just to show you, here's one of the patients. You don't see an atrial beat, got a tachycardia going. 2 hours after you get the drug, there's the nice P wave and a nice normal rhythm. So, if you get a chance to go to Bilbao, see Frank Gehry's work, greatest museum ever built. That's what we're shooting for. So let me just finally say a little bit about predicting cardiac safety. I think this is the way to go. So if you want to do it with patch clamp, you're going to be able to do a one compound in a third of a day. TAT means turnaround time. You can decide whenever you're doing it, if you get the HERG signal, you stop. If you did it here, you stop. If you do it here, you stop. If you do it here, you stop. And you're going to have an error rate that's about 25 to 30%. If you do mice, you can do 10 compounds in the same amount of time. Therefore, if the turnaround time and the labor involved, which is the most expensive item, then you know that it's not going to be more expensive. And so if it's HERG selective, quit. But if it's got CAV and NAV offset, you should continue. You can do a couple more channels, the big 5 I call them. Again, it's very, very fast. And so at the end of the day, you will have a mechanistic understanding of what's going on for Torsades-de-Pont, and you'll have a much lower error rate. So just to conclude, mice is 3x better than HERG as the predictor. It halved the false positives and reduced the false negatives 6-fold in our data set. And the TATs are very much faster when you do this in automated mode. So what about SIPA, the comprehensive in vitro proarrhythmia assay, which we heard a lot about today? I'm with you guys. I think this we've already shown. That's not to say you have to do it the way we do it. It needs to be standardized. I'm not disputing that for 1 second. And this, in my opinion, This is a walk in the park. And this one's not a problem. I mean, I really don't see that as an issue. But this is going to be difficult. And I think this is where, I understand the rationale. I do. But this thing is just loaded with landmines. So, I think there'll be a lot of discussion about, the SC-derived cardiomyocytes, because they're not perfect. And, if you can figure out, okay, so let's say you get the EAD. I didn't get a chance when I showed you that slide with the ECG and all that stuff. But I think sometimes we don't keep in mind what the hell is really going on in the ventricle. And the ventricle just doesn't sit there and do that. The electricity goes down, and it comes out of the AV node, and it goes through the conducting system very quickly, because you know what? It has to hit the papillary muscles first, because the mitral valve has to close. The aortic valve's already closed, the mitral valve has to close. And then the next thing that has to come in is that the apex has to move up to the base, because you want this thing to be a sphere in terms of efficiency of contraction. And then you have to bring into play the circumferential muscles, which will now make the isometric tension exceed the aortic pressure, and you'll get ejection. Now, activation requires propagation, but remember, repolarization is not active. Repolarization depends on how those cells have been set up. So why do you have an upright QRS and an upright T? The reason you have it is because for depolarization, in general, it goes from inside out. And repolarization, for God's sakes, why doesn't it go from inside out? It doesn't, because the epicardial cells have been arranged in such a way that they repolarize faster than the endocardial and myocardial. So it goes from outside in, and so they're both upright. The great designer in the sky mystifies us once again. So this is what I see, Gary, for what you're in for. Not only is it appropriate to have the cat walking by all these guys, but for you to get through number 2 is going to be like herding cats. So good luck. And with that, I'll close. You've heard me mention, I don't have a picture and I don't have the names, because you don't pay much attention to them anyway. But I've been lucky over the years to have had really wonderful collaborators. I mentioned Dave Rompey. I've talked about Yuri Kurashev, who's with us now. Most of the guys at ChanTest with me have put up with me for, believe it or not, something like on average 20 years. So, we have Yuri, we have Glenn, we have Carlos, we have Jim Kramer, we have Andrew Bruning-Wright, we have Jessica Brimacombe, we have Chris Mathis, who's really understands this stuff both from the customer point of view as well as from the scientific point of view, and probably some other names I've left out. But it really has been, I've just been lucky to be around and participate in the drive. So thank you very much.